So we will continue the post-lunch session from now. So Dr. Hajra, it's all yours. Please start.
Thank you. So, as I was talking about, we have discussed three type of modeling methods. One is homology modeling, then is gen threader or fold recognition, and another was ab initio. But you know, now there are packages appearing where it is not one method or the other, rather the combination of all these methods which goes to success. And two such methods are ITASR and Rosetta. So ITASR, it is based on multiple threading alignment. It's also called Zhang server. Why Zhang server? Because the owner of this server is Yan Zhang. But there is an interesting story which is very new about this, both the groups. What Yan Zhang did, all the PhDs and postdocs came to his lab. He have given them problem which they have carried out to their own lab. What I am trying to say is, let's say I have a lab and there are 10 students. Tomorrow that 10 students have one problem, taking to, on the same topic, taking to 10 different labs having 100 students. They goes to another lab having 1000 students. In that way, Zhang server and Zhang lab is a really big user lab, and they become #1 server for protein structure prediction in CASP experiment. What is CASP experiment? CASP experiment is very interesting. First, let's see the CASP ranking. You see Zhang server is on the top, but you also see there is something called Quark. I'll talk about that also later. So ITASR has been consistently ranked as the top method in CASP, a community-wide experiment to benchmark the best structure prediction method in the field of protein folding and protein structure prediction. CASP takes place every 2 years since 1994. So CASP is, you could say, Champions Trophy, World Cup of this structure prediction. Every 2 years it happens, and in these 2 years, how many problems you have solved, based on that, how efficient your program is, how many user you are having, based on that you get points. If you don't know about CASP, I could give you one reference which I will talk about also. You don't know CASP, but you all, I hope, know about AlphaFold, right? AlphaFold came into limelight after becoming top in CASP by putting Zhang server behind. So I'll talk about them. So this is the record. It is 2006, 8, 10, 12, 14, 16, 18, 20 there, becoming the top one. So how the program run? The input mandatory, you have to give amino acid sequence with the length from 10 to 1500. Think about, this is one of the things you should consider, a very interesting one. If you have a protein with more than 1500 amino acid, you don't have this program open for you. And if people know about proteins with medical importance, there are many proteins which are working as a platform, and they are mostly on 5000 amino acids. They would not be predicted using this server. Mandatory, you have to give this. Optional, you can provide optionally restraint and template to assist ITASR modeling. So when you are doing modeling, you make a 3D structure, so there is restraint between bonds. You could have include that, and you could have provide the template which would be used to model the program. You could provide contact restraint, you could provide distance map between the atoms which are in space, inclusion of special templates, exclusion of special templates. You know that some templates are coming in the search, but you feel that they are not good. So you could have exclude them, and you could also include secondary structures. The output, you get structure prediction and function prediction, because that's what we are doing. In structure prediction, secondary structure prediction, solvent accessibility prediction, top 10 threading alignment from Lometz, top 5 full-length atomic models, top 10 protein in PDB which are structurally closest to the predicted models, estimated accuracy of the predicted model including a confidence score of all models, predicted TM score and RMSD root mean square deviation for the first model, and par residue error of all models. And B factor estimation. B factor is the thermal factor. B factor says the disturbance of the amino acids inclusion in a protein. Then for functional prediction, you get enzyme classification and the confidence score. How confident? These are gene ontology terms and the confidence score, ligand binding sites and confidence score, and image of the predicted ligand binding site towards assisting drug discovery. So how they work? This is the schematics. I would like to try to explain all the steps. The first step is threading. Remember threading I talked about? So you have sequence, okay? You chop the sequences into small fragments, okay? Then all these you map with the existing fold. Is it matched there? If it is matching, then you get the folding information coming from them. Then once you get them, you get a collection of templates. What happened actually? You basically get few of the secondary structures. Let's say you have one helix, one beta strand, two beta strand, second helix, like that. Now you will go to connect them, right? So let's say this is a alpha helix, this is a beta strand, okay? You could connect this with this, this with this, this with this, this with this, right? All possible connections you are allowed to. But then you develop a ab initio program. What type of ab initio program? Remember, you have a equation needed and you have to screen for a certain criteria. Kaise? So you have all those connections, right? Now with all those connections, some connections are right and most of them are wrong, right? When they are wrong, then there is a chance of steric clash. You understand? So you make all the fragments connected to all possible direction without maintaining any rule. So one set is correct, rest of all set are wrong. Among those wrong of them, most of them will show steric clash. Your ab initio program is based on finding steric clash. So if there is a steric clash, all the structures would be eliminated. So all your maximized wrong structures would be eliminated using ab initio scoring. Then you get mostly non like steric like connections, right? So that's what here is. Now you will do, so this is, see, you did threading, you did ab initio. Two process you did. Third process is even interesting. Imagine a page in front of you, okay? And you take a structure, any structure, okay? And rest of the structures are there. You take one structure and align them. Based on this alignment, you will be positioning. So what I mean is, think about, this is the 2D page, okay? You are happy. Now, any point, you take any structure, put it here. Here you put, then other structure come, based on their relation, put here. all the drops are coming 2D. That drops you could see here. Everyone understand what is happening? You have a 2D sheet, you put one structure, and now based on their structural relation, you put them there. So the structures which are correct, they are many in number. So they will be in one place. So look at these. Few places you get them dense, intense. So here we get one place intense. We make a circle around that. And we get to the center of that. Is it a perfect circle? No. So the center will be perfect? No. That's why we call it centroid. Are you getting it? So we make many drops. The place where maximum number of dots are accumulated, we focus on there. We are not looking at others. What we did now, we draw a circle around that. So dots are random. So the circle would not be perfect. So if you get a center of the circle, that would not be a perfect center. That's why we don't call it a center, we call it centroid. And it comes through a clustering, so it cluster centroid. Okay. Now, because it is not perfect, so there would be not one structure, there would be a collection of structures. Right? So from here, from the cluster centroid, we get some collection, and we do energy minimization, we get the one structure. And up to now it was backbone development. So when we get the backbone, we add the rotamers. And now we get the final model. Now one very interesting thing, if you go back to the clustering, you see that the cluster we have taken, beside them there are smaller clusters, right? That's what perfectly happening to all the proteins, because it should not be one conformation of a protein. So the dense cluster actually representing the best conformation, thermodynamically most stable conformation, but there are other functional conformations. When it go to perform a reaction, when it go to bind a protein, it might open up. So all those conformations you could see many cluster centers. Okay. So in that way we could develop the model, but this is the basic one. So now we are going to go for complex. So in addition to the classic ITASR pipeline, several approaches were recently developed and integrated into ITASR to enhance its ability of structure modeling for distant homology targets. What are these? So 1990 to 2003, when human genome projects are happening, a lot of new protein came. That time the people who are doing modeling was not as unlucky as you guys. Now the easy fruits are gone, right? So if now you want to do modeling, you have to take tricky proteins. These tricky proteins are called distant homology. Like the homology is not very common, so you cannot do homology modeling. That's the thing. So they are doing a lot of things to work on them. What are they? The top models generated by the quark ab initio folding were merged into the threading template pool, which were used as the starting conformation. Remember I told through ab initio you could do quantum calculation. Through quantum calculation you could make peptides. Now if you look here, here you are making small fragments through threading. Why not that threading pool you add the structures you are getting through this quark? You all get my point? you cut into small pieces and you find matching of the fold. Now, that is through threading. Now through ab initio program, you do the same thing. You are doing the quantum calculation of small fragments. So in both the way you get small fragments. So why don't you merge this pool to the already existing pool? So you will get more structures, more candidates. So that's what here in quark ab initio we are doing. Since the hard targets generally lack global templates, the sequences are broken into segments of 2 to 4 consecutive secondary structure elements, which were then threaded through the PDB by the segmental threading tool Segmar 9 to identify supersecondary structures. So you have a big protein, okay, and big proteins are having domains. Domains are connected by non-structural elements. So you identify those domains. You identify them. Supersecondary structure. What are supersecondary structures? They are little more complicated than secondary structures, like 1 alpha helix, 1 beta sheet, 1 alpha helix. Helix, strand, helix. You know a very famous like supersecondary structures, helix, loop, helix, which binds to DNA. So those type of things are available. So you could use that to divide them and utilize them there. SVM, SIC and SPCon. So you are using this. SVM is support vector machine. This is a machine learning based algorithm. You use machine learning to develop the residue contact map. Residue contact map. What is residue contact map? The residues are interacting. I am finding a profile by using a model where I am studying the common interaction between the amino acids in different proteins. So I have studied different proteins. I have seen that a arginine forms salt bridge with a glutamate or aspartate. So I know this knowledge. So I apply that knowledge of interaction by finding the amino acids and develop a AI based model. For multiple domain proteins, the protein with many domains, Threadom is a tool. It is used to predict the domain boundary and the linker region, like where the domain ends and how it is linked by non-structural elements. The ITASA suite is a downloadable package of stand-alone computer programs developed by Yang Zhang Lab for protein structure prediction and refinement and structure-based protein function annotation. Through the ITASA license, researchers have access to these programs: ITASR, Coach, Cofactor, TM-Site, S-Site, Lomets, Master, Speaker, HAD, ETSUB, Mod Refiner, NW Align, PS Spread Library. All those are programs in their package. Okay, I have explained them. I will send my slides so that you all will receive all those lists and could use those programs. This is Yang Zhang, and see the structure comparison. This structure this is a structure. This is determined by experiment, and this is the structure they independently made by modeling. So what is the future target of ITASR? Despite the success, all the success you have seen, significant unsolved problem remain in protein structure prediction, which will be target of ITASR in coming years. How to build structures of experimental resolution below 1 to 2 angstrom useful drug screening when homologous templates are available. So they want to make higher resolution structures available for the user. How to identify distantly homologous templates with accurate query template alignments. So how to bring that where the relation is not very straightforward, the relation is tricky. How to bring them also in down. How to fold protein, especially beta proteins, with correct topology by ab initio modeling when there is no template. How to fold membrane proteins. So why membrane protein is like separately mentioned? So there are two type of proteins I told. One is cytosolic protein, right? Cytosolic proteins are soluble, right? To solubilize what you need? Water and buffer. So together water is free and buffer is probably 50-60 rupees. So you could solubilize a protein with 100-200 rupees. But the same thing is not true with membrane protein. Why? Because soluble protein are soluble in water. You know what is water, but you don't know what is membrane. And membrane is not one thing. So exactly what is the membrane component? What are the closest component where the protein would be solubilized? You know, the membrane protein labs, they do a screening, and the minimum cost of that screening is 50 lakh rupees. So that's why membrane proteins are very challenging in comparison to soluble protein. Nowadays, thanks to cryo-electron microscopy, we are solving some of the membrane protein, but still they are challenging. So that's why ITSR keep it also in their vision. And how AI deep learning could be incorporated in the structure prediction. And when we talk about AI deep learning, we are coming to the last segment of the modeling part, which is AlphaFold. And AlphaFold is, as you all know, famous for suddenly people thought that every problem is solved through AlphaFold, which is not true, but it's a good start. And this year, Nobel Prize is given. Now, Nobel Prize is a big thing, and I should not comment on that, but if I understand some amount of this, I think there is a tendency for the Nobel committee to nowadays bring people who are doing like same work. Like this year in chemistry, they have given prize to AlphaFold creator, the DeepMind people, and David Baker. Now nobody question about David Baker, but I think for AlphaFold, it's much earlier time. They should have given, some more time to prove and come up with much more reliable structures. Anyway, so AlphaFold's development marks a significant milestone in computational biology, beginning as an ambitious research project within DeepMind and evolving into a world-renowned protein structure prediction tool. Here is a brief overview of its history. Initial development 2016 to 2018. See, that's what I also told. Here, history is 2016 to 18, right? That's the interesting part, right? So, DeepMind began exploring protein folding with the goal of solving one of the biology's most challenging problem. The approach initially used machine learning and neural networks to capture complex protein sequences and their possible structures. By 2018, DeepMind's early AlphaFold prototype participated in CASP13, which we talked about, a competition where computational models are tested against experimentally solved protein structure. AlphaFold saw good performance, demonstrating that its approach to protein folding had significant potential. Now, AlphaFold is improved and come up in the name of AlphaFold 2. In 2020, DeepMind unveiled AlphaFold 2, a much more advanced version that used a breakthrough architecture, including the EvoFormer module. This is the actually game-changing thing, EvoFormer, where they are actually taking the concept of evolution. How nature is evolving, they take the concept and develop a model which take the way nature is evolving in a much speedier way. So including the evoformer module and attention mechanism within a transformer based neural network. AlphaFold 2 was tested as Cas14, which achieved astonishing accuracy predicting protein structure with atomic level detail. This performance was recognized as a substantial breakthrough, offering predictions nearly as reliable as experimental data, yet orders of magnitude faster. Public release and impact. DeepMind made AlphaFold's code and model weights open source in 2021. So they make it open to public. Then they released the AlphaFold protein structure database in collaboration with the European Bioinformatics Institute, or EBI. So this database initially contained over 350,000 protein structures and rapidly expanded to over 200 million by 2022, covering nearly all known protein in major organisms. The impact is such big that, you know what is this? This is the front page of Protein Data Bank. Okay. So Protein Data Bank is a database which is mainly developed because of experimental structure. But here you see, and this change happened only in last year. If you go to PDB earlier than last year, you have never seen this thing, computer structure models, or CSM. So this is the structures of PDB, but you see the beaker is representing them and the computer is representing the computer model structure. Ongoing development and global impact. The release of AlphaFold transformed fields like structural biology, genomics, and drug discovery. Its models are integrated into platforms like UniProt, Ensemble, allowing researchers across disciplines to access detailed protein structure for numerous applications, including addressing disease, improving crop resilience, and developing biotechnological solutions to environmental issues. So they are not only predicting the structure, but making it in such automated on click that a person who don't know about anything, they could also look at and take the advantage of structures. AlphaFold's success underscores the potential of AI in scientific discovery, bringing the protein folding problem, once considered nearly unsurmountable, within reach and opening doors to further advances in medicine and biology. So UniProt, I talked about this UniProt. UniProt is a comprehensive publicly accessible database of protein sequence and functional information widely used by researchers in bioinformatics, structural biology, and biomedicine. Created through a collaboration among European Bioinformatics Institute EBI and Swiss Institute of Bioinformatics SIB and the Protein Information Resource PIR, UniProt provide detailed curated information about proteins, including their sequence, functions, structures, and interactions. So about one protein, if you know the UniProt ID, you will get each and every information available in the world. I talked about UniProt ID. You see, you get 6 IDs, like 6-letter number, or you get 10-letter numbers. And if you go to UniProt ID, you will see one very funny thing. So this is the model made by AlphaFold. Okay. Now, if you see, you will be seeing that this part is very good model, right? According to the color code given by AlphaFold, and this part is very low, right? Any guess why? When they are, modeling one part, majority of the part so nicely, why a certain part is so badly modeled? Any thought? No? Try at least. So that should be, but that's one of the flaw of AlphaFold. So, you know, when you see any protein, you will see that the protein have a signal sequence, and signal sequence is nothing to do with the structural part, because the signal sequence is to reach the protein to its destination and then chopped out. So this is not part of the protein. Somehow this is still not included in AlphaFold. So those are my points, that AlphaFold have to be, you know, mature with many small things. So I have like made a model by myself, and you could easily see the difference between these models. So that's the thing, like those are the areas there, you know, could be working well. So AlphaFold have multiple sequence alignment. It starts by analyzing and multiple sequence alignment, a comparative matrix of similar protein sequence across different species. This process helps in understanding the correlation between the evolutionary process. This alignment hints at the likelihood of certain residues being close to each other, especially based on evolutionary covariation. So they do that alignment so that you see that there is a very important position, and that important position is conserved, or a very important position and it is very much varied. So those are very important clues about evolution. Embeddings and evoformer module. The multiple sequence alignment data is transformed into embeddings, continuous representation that the network processes. In the evoformer, these embeddings are refined through iterative updates that analyze both residue pair interaction and structure. Remember, as I say, if you have arginine or arginine alanine, there is a lot more difference, right? So just one pair of residues could make difference. So that they are picking up here by optimizing the pair of interactions, pair of amino acids. The system learns from the MSS sequence patterns, then updates its 3D structure hypothesis iteratively using feedback loops to increase the predictions accuracy. Transformers attention mechanism. The AlphaFold's evoformer relies on a transformer architecture with an attention mechanism which prioritizes specific part of the multiple sequence alignment and current structural hypothesis. So how the structure is developed and how the multiple sequence analysis showing that some parts are conservatory, very like authentically very conserved, and some part are very variable. This allows the system to focus on parts of the sequence critical to structure formation, producing increasingly accurate atomic details of protein model with each cycle. Iterative refinement and output. Finally, the iterative cycle of updating the sequence and spatial relationship in AlphaFold's model lead to a near complete structure assessed for accuracy against known structural databases. And this combination of evolutionary data, structural hypothesizing and feedback enables the AlphaFold to predict protein structures accurately and efficiently, typically within minutes. I am mostly end of this part, and I will introduce you to Institute for Protein Design. This is the institute from where David Baker is hailing. So this is David Baker, the Nobel laureate this time, and they have developed a organization which is called IPD, or Institute for Protein Design. And you could see the huge, funding they are getting, and the executive function of naturally occurring protein is to solve the challenges faced during evolution. However, they face challenges today that were not faced during natural evolution. The goal of Institute for Protein Design is to develop and apply method for designing a whole new brand of synthetic protein. What that means? That means incorporating unnatural amino acid. You know, how many natural amino acids are there? 20. How many unnatural amino acids are there? Impossible number, right? So the moment we allow unnatural amino acids, our synthetic capability would be going from this to this. So that's what they are doing. They are coming up with new materials, new vaccines, everything, and more importantly with folds which are not in nature. To achieve the previously mentioned goals, the Institute for Protein Design, established in 2012, the protein design requires high-level expertise and talent in computing and software, biochemistry, genome sciences, biological structure, pharmacology, immunology, and other basic science disciplines, as well as clinical medicine. In one word, a completely interdisciplinary setup. So that's what they did, from a structural computational department to a true interdisciplinary setup. And they have already developed so many startups. It is an initiative to build up a wonderful team using faculty, scientific staffs, postdoctoral fellows and graduate students in University of Washington, as well as developing partners from collaborating institutions, innovator networks, and from the computer and biotechnology industries, bringing extraordinary expertise to bear on a singular focus to advance the potential of protein design. Over the past 16 years, UW researchers have made significant progress in protein design and protein structure prediction, developing the world-leading Rosetta software. So, I talked about ITASR. The second one was Rosetta. That is what David Baker and their team developed. Over this period, UW scientists have developed methods for designing proteins with a wide range of new functions, including catalyst for chemical reaction, HIV and RSV vaccine candidates, flu virus inhibitors. The IPD integrate these strengths in protein design with Seattle area expertise in biochemistry, engineering, computer science and medicine, and leverages the exceptional Seattle strength in software industry. Even though design a protein remains a challenging task due to the large sequence space that requires sampling, few landmark development had changes the way how we looked at it. So remember I told we have the huge amount of sequence space. It is really impossible to merge the sequence space with the conformation space. But these approaches have really elaborated the idea of conformational extension. The number of gene sequence enhanced astronomically following up the success of Human Genome Project. Next generation sequencing techniques are not providing us the sequence of the gene or protein, it also providing genetic regulation, expression patterns, effect of factors influencing gene, protein expression. All of these are invaluable in correlating structure to function. Though much smaller in comparison to sequence, the number of resolved 3D structures also increasing day by day. So the 5 grand challenges I'm just going through: building better vaccines, so protein modeling has become an indispensable tool in structural biology, offering critical insight into the structure-function relationship of protein. By leveraging the computational methods, researchers can overcome the limitation of experimental techniques such as the 3 techniques we are talking about. Methods such as ITASR demonstrate the value of integrating template-based approaches with ab initio refinement for functional annotation and structure prediction. Tools like AlphaFold have set a new benchmark for structural prediction accuracy, enabling the reliable modeling of proteins even in the absence of homologous templates, where the templates are distant homology. Each protein modeling technique, whether homology modeling, ab initio, threading, series unique proposes emphasizing the importance of selecting the right approach based on the biological question. Like techniques are in your hand, which techniques to avail, which process to take on, it is depending on what is your problem, what is the problem you are looking for, and how you think about that. Computational protein design offers unprecedented opportunities to engineer novel biomolecule for specific applications, including enzyme optimization, drug development, and therapeutic protein creation. Despite significant progress, modeling techniques still face challenges with intrinsically disordered regions. There are some of the protein supposed to be folded, right? But there are, in normal situation, but there are some proteins which are intrinsically disordered. They are getting ordered only when they get their proper partners. So those proteins are very challenging to handle. Experimental validation remains critical to ensure the accuracy and functional relevance of computational models. Integration of AI-driven techniques with molecular dynamic simulation can enhance the study of protein flexibility and interactions. Expansion of experimental data sets like cryo-electron with further refined predictive models and expand their applicability to previously inaccessible protein families. In conclusion, protein modeling is a rapidly evolving field that bridges computational innovation and biological discovery. By integrating the predictive tools with experimental validation, researchers can accelerate advancement in understanding protein function, designing novel biomolecule, and tackling complex biological challenges. So I finish here this part. Now, do you have computers with you? So ওইটা দিয়ে দাও ওদেরকে, ওই যে সিকোয়েন্সার শিবাশিস, ওই এটা দিয়ে দাও সবাইকে। হ্যাঁ হ্যাঁ কিছু বলতে হবে তোমাকে? So let's start the hands-on session. So in this session, I will try to, like do things. First, I will talk about the relation between gene sequence with the protein, very basic. And from there, I will go to sequence analysis with different processes. Then we will go to model. After that, we'll talk about the PDB format and the coordinates, how the coordinate is there in the file and what other things are there in the .pdb file. I also wish to show two visualization softwares, but one, the code is not downloaded here, so I think we'll finish by showing PyMol. Okay. So gene sequence, we talked about a lot with genome data. The genome data comes with the output of gene sequences, and when you have gene sequence, you go to the codons, you will get the protein sequence. There are many tools, but I will be giving you example of one of the good tools so that you could have these slides and go there, just click it and do the process. Okay, because I don't know how many of you following me, so I kind of trying to demonstrate it in a way, it is a kind of tutorial. Okay, so you go to this XPASI tool. XPASI is the server which called translate. So in the translate tool, when you provide your gene sequence and put translate, you would get the protein sequence. Easy, and you will see that the first one, the first codon is ATG. You go here, you see that ATG is methionine, here it is AUG, and in protein you see methionine. Okay, so you could manually do that. At the beginning, many people used to do this change manually, but gradually the tools are developed. Like here, it is a tool called translate. Okay, the reverse process, it's not easy. From gene to protein is easy, but protein to gene, because there are number of codons, so that's why it would not be straightforward. Remember that. And whatever it is, be it gene or protein, when you are going to work with them, you have to keep them in a format which is universally called FASTA format. So in the field of computational biology, the FASTA format is a text-based format for representing either gene nucleotide sequence or amino acid protein sequence, in which the nucleotides or amino acids are represented with single letter. This format, thank you, also allows for sequence names and comments to precede the sequence. So you could put the name and comment so that you could remember and not mix up with other sequences. The format originates from the FASTA software package. The FASTA format, it comes through FASTA software package, but has now become a near universal standard in the field of computational life science. So initially there are many formats, right? One of them was FASTA, but everyone liked this format, so they start following it. So now FASTA become a universal format. The simplicity of FASTA format makes it easy to manipulate and parse sequences using text processing tools and scripting languages like R programming language, Python, Ruby, Perl. Parse is thing when you have the thing process. It could be taken in a logical way. Let's say I don't know your names. So if I have to call someone, I will be like trying to call, hey, hello, hello, and then right to you, left to you. So this is a illogical process. This is not a logical process. To develop a logical process, you will be developing something which is called parser. If you have a parser, like here, if you all hailing from one class, you all have a roll number. So roll number is the parsing ability here. So every data now is going to be like according parser mode. The original FASTA Pearson format is described in the documentation for FASTA suite of program. You could search. It can be downloaded with any free distribution of FASTA. In the original format, a sequence was represented as a series of lines, each of which was no longer than 120 character and usually don't exceed 80 character. This probably was to allow for pre-allocation of fixed line size in software. At the time, most users relied on Digital Equipment Corporation or DEC, VTT20 or compatible terminals which could display 80 or 132 characters per line when the FASTA was developed. Most people preferred the bigger font in 80 character mode, and so it became the recommended fashion to use 80 character or less, often 70 in FASTA lines. Also in width of a standard printed page is 70 to 80 characters. So this is not something as a rule. This is people use it and they get comfortable to use it. As a result, 80 character become the norm. The first line in a FASTA file started with a greater than symbol. Less frequently, a semicolon was taken as a comment. Subsequent lines starting with a semicolon would be ignored by software. So if you want to put something, you put semicolon. This would not be considered as the sequence part. Since the only comment used was the first, it quickly became used to hold a summary description of the sequence, often starting with a unique library accession number like UniProt number, and which with time it has become commonplace to always use the greater than for the first line and not use semicolon on the comments. Following the initial line used for a unique description of the sequence was the actual sequence itself in standard one-letter character string. So you have the greater than name of the sequence, then the sequence, one-letter sequence. Anything other than valid character would be ignored, including the space, tabulator, asterisk, etc. It was also common to end the sequence with a star character in analogy with use in PIR format sequence, and for the same reason to leave a blank line between the description and the sequence. So if you see, there are three sequences I have taken two groups, one group 1 and group 2. I hope you get your sequences, all of you. So I have given a bunch of sequence so that you could all pick up one sequence. Okay. So there it is written, TEM1, the UniProt ID is given, SHB1 UniProt ID is given, BEL1 UniProt ID is given. If you go to uniprot.org and search with this UniProt ID, you get the details of the protein. So, I have picked up these three proteins. These three of them are class A beta lactamase. This is what I work. And in group 2, I pick up 2 metallo-beta-lactamase, NDM1, which is called New Delhi metallo-beta-lactamase. Now, I put it purposefully. I just want to show you that, sometimes you see things you don't like, so probably you will protest about that. We have seen disease occurring from countries and all specifically, right? But we don't call them on the name of the country. It is defamation of the country, right? But that same thing happened when a beta-lactamase, which was found from Gangaram Hospital in Delhi, they have named it as New Delhi metallo-beta-lactamase. This should not be done, but that time there was no scientists working in those fields, so they probably don't contest against that, and this name exists. So this is a metallo-beta-lactamase named on a city, Delhi. So sequence alignment. So once you get the sequence, you know how the sequence have to be written in FASTA format, the standard format. You go for alignment. Sequence alignment is a fundamental computational tool in bioinformatics that compare biological sequence, be it DNA sequence, RNA sequence, protein sequence, to identify the region of similarity, which may include functional, structural, or evolutionary relationship. So it could have been structurally similar, it could have been functionally similar, or it could be in terms of evolution similar or having a trend. Key concept. There are many different concepts in the alignment. Pairwise alignment, which compare 2 sequences. Multiple sequence alignment, MSA, align 3 or more sequences to highlight conserved regions across multiple sequences. So if you have only 2 sequences you want to compare, you will be going for pairwise alignment. If you go for more than 2 sequences, you will go for multiple sequence alignment. Alignment categories, global alignment. Attempt to align the entire length of 2 sequences. When we are comparing a country with another country, the entire country, or we are comparing one place with other. When we are comparing one place with other, it is called local alignment. When we are comparing the whole thing, it is called global alignment. Global alignment is the attempts to align the entire length of 2 sequences. It is most effective for sequence for similar lengths. The algorithm is Needleman-Wunsch algorithm. I am not going into details, but I will show you what to do. Local alignment, it identifies the regions of high similarity within the portion of the sequence. Smith-Waterman algorithm. So those are processes, but when we do, we actually try to optimize. You don't go for a one-shot thing. And when you optimize, you try and you go for better. That is called heuristic method. So it's faster but approximate alignments for larger databases. Example is BLAST. Remember, in time of protein sequence modeling, we are taking the help of BLAST. So this is the site, this is the link through which you go to BLAST, and this is the page. You would put your sequence here, and when you put your sequence here, you put BLAST. But in between, there are some interesting ones. You see here, can you read that? Non-redundant protein sequence. When you use non-redundant protein sequence, when you are searching for a unknown sequence, you don't know with whom your sequence will show similarity. You get a new bacteria, you get a new plant, you get a new, something, and you get the gene sequence. So you want to see that with which protein, what type of protein your gene is aligned. That time you will go to this inner database. But if you want to model, then you use the database, PDB database, because you have a sequence, you are aligning it with a sequence, that sequence have a structure. Which sequence have a structure? The sequence which is deposited to protein data bank. So you use that as a data bank. So BLAST is simple, but BLAST have many options. You have to understand what you are doing. The problem of bioinformatics is when I will say it will be nice, but you have to do it. When you do it in hands, you will get simple problem, difficult to solve. More you play, we say in bioinformatics, more you play, more you practice, more you look by yourself, better bioinformatician you will be. Today I am teaching it. If you start asking question about everything, there will be things coming out I don't know even. It's possible for everyone. So if you want to be a good bioinformatician, you click on all the options. Even the options are not required, and try to know about them. Okay. So you put your sequence and you do the search, and see, you get a search like that. But if you use PDB, in that case, you see here there is no structure, right? But if you use PDB, you get this PDB ID. Everyone, can you see that? Can you understand? So two searches I have shown you here. In one, I put NR database. So it shows 100% identity because I know the gene. I know it is TEM. TEM is in the database. So it is showing 100% identity with TEM. But when I am giving structure, might be the sequence is known but structure is not known. Fortunately or unfortunately, TEM have structure, so you get a 100% match. Otherwise, you could directly get maybe 40% match. So those are tricky thing you have to look at. Then there is heuristic alignment. Then there is progressive alignment. Progressive alignment is a heuristic approach. Everyone where you try to optimize through computation is a heuristic, is a process done by computer. So you get it, you get 70%, it will go to getting 80. You get 80, it will get to getting 90. In that way, it will be doing better and better. That's called heuristic. So progressive alignment is a heuristic approach used to align multiple biological sequences, DNA, RNA or protein. It is one of the most common method for multiple sequence alignment. BLAST was for pairwise sequence alignment. This is for multiple sequence alignment and is based on constructing the alignment progressively, starting with the most closely related sequences and gradually adding the others. Example is Clustal W. This is the link of Clustal W. You click, you will go there. This is the page. Here you will be putting the sequence. But here you need multiple sequence. So I put 3 sequence because you need multiple sequences. And see here, different formats are given. Though we are doing Clustal W, but there is PRRN, there is MAFFT, there is ETE3. All are different type of, format of doing this. Okay. So we use this link and you get this alignment. These colors are showing when there is exactly same amino acid or something like that. And because these three I told coming from same group, class A beta lactamase, and they are quite similar, so you see the reflection, many residues are identical here, right? Then another alignment sequence you do, which is multi-align. When you do cluster W, you get a outcome like that. Whereas when you do multi-align, what you see properly, you will see that, here you are getting only sequence, right? Now carefully look at, in multi-align, you also see the secondary structure. Can you see it? The helix, the seat at the top. This is the helix. This is the beta seat. Can you see? Beta 2, then alpha helix. So it not only calculate the alignment, it also gives you the primary calculation of secondary structures. This is between group 1 and group 2, because there are dissimilarities between group 1 and group 2. You see the red color is now not as much as you see, right? So that's the direct effect. This is also the multiple sequence alignment through multi-align. Then we will talk about another software which is very interesting, Conserve. Conserve is a bioinformatics tool designed to analyze the evolutionary conservation of amino acids or nucleotides in a protein, DNA, RNA. So this one will tell you, you have a protein, this protein have residues, but which residue is whole through, like between the homologous domain in the world of protein, they are conserved or not. Very nice finding. It maps these conservation scores onto the 3D structure of the protein or nucleic acid, providing insight into functionally or structurally important regions. This is the link and this is the server, Conserve server. So this is the results. First thing you see, I have not given them the model, right? So they by themselves automatically calculate model. How they automatically calculate model nowadays? Using AlphaFold. Everywhere AlphaFold is integrated, right? So the same mistake is there, see. Can you see? This extra region which I told it's wrong, they have given it there. But forget about that, they have many interesting color. You see this color? From blue to maroon, the blue, this dark sky blue, which is core 1, when this comes, it is most variable. And when it comes to this 9 score, this maroon, it is most conserved. So now you could see here which residues are conserved, which residues are variable. Also, you see different numbers, like different letters. You see E. E is an exposed residue according to neural network algorithm. So a neural network algorithm is applied to them and it shows that the E are exposed residue. They are coming on the surface. B are buried residues. F are predicted functional residues. S are predicted structural residues. And X are residues about which they don't have sufficient information. So more X you get, more newer structure you think you are, or newer protein you think you are having. So these are the overall comparison between different proteins. So you see different residues, how they are aligned, and they are different residues we are taking, and you see the color spectrum, how they are differentiating. Now we are going for modeling. So I have given sequence, you could have used those sequences. So we are using Swiss model. Swiss model is an automated web-based tool for homology modeling of protein 3D structures. It enables the researchers to predict the 3D structure of protein based on its sequence and homologous protein structures available in the database. Homology modeling is particularly useful when you don't have structures. We already have talked about that. This is the web server. The requirement is target sequence. The protein sequence should be in FASTA format. That's why I started with FASTA format. Optional, a specific template structure or custom alignment. You could provide a template or you could not provide a template. This is your choice. Steps: first you identify the template. You search for homologous protein with known 3D structure in protein data bank. Select the best template based on sequence similarity, coverage and experimental resolution. Template alignment: you align the target sequence with the selected template. Then you do the model building. You construct the 3D structure by transferring the spatial coordinate from template to the target. You refine the model to account for differences between the template and target sequence. This is the page of the Swiss model. You could start modeling by incorporating residues here. And when you do the model, how you judge the model? You judge the model based on two factors. One factor is GMQE or Global Model Quality Estimation. So it is a scoring matrix used in Swiss model to evaluate the reliability of the predicted protein structure. It provides a quantitative estimate of the expected quality of a homology modeling before the actual 3D structure is built. So it will give you many options and their scores. Based on that, you could choose the highest or high GMQE model. GMQE ranges from 0 to 1. Higher value indicate greater confidence in the predicted model. High GMQE means greater than 0.7. It is a reliable model. Medium GMQE 0.4 to 0.7, moderate reliability. Low GMQE less than 0.4. GMQE integrates sequence identity. It is based on higher sequence similarity between target and template, improves the GMQE. Coverage: the alignment coverage between the target sequence and the template affects the score. Partial coverage reduces the GMQE. Template quality: high resolution structures from the PDB yield higher GMQE score. Template with structural errors or low experimental resolution lower the GMQE. Another one like GMQE is Q-mean. Basically you have to collaborate between the scores of GMQE and Q-mean. Q-mean is qualitative model energy analysis. Q-mean is a scoring system used to evaluate the quality between 3D models by comparing them to experimentally determined structures. It is widely used in homology modeling workforce such as Swiss model to provide a quantitative assessment of the reliability of a predicted structure. There are key features. Composite scoring system combines the multiple energy terms to assess both the local and global structural features, evaluates the agreement between the predicted model and structural characteristics typically observed in high quality experimental structures. Normalized G-score, the Q-mean G-score allows the comparison of the predicted model's quality with experimental structures of similar size. The Z-score range close to 0. Negative Z-score indicate a model significantly deviating from typical high quality structure. It is applicable to protein of various sizes and complexities. Provides both global and local quality assessment. How Q-mean works? Q-mean combines several scoring terms into a single quality matrix, local quality terms, torsion angle potential, secondary structure packing, global quality term, solvent accessibility agreement, interaction energy, normalized scores. All the terms are combined and normalized to generate the Q-mean score. The Z-score indicate how the model compares to a database of experimental structures. Interpreting the Q-mean score, the Q-mean score is a composite score. Higher value indicate better model quality. No fixed range is there. It depends on the size and nature of the protein. The Q-mean Z-score, it indicates how far the model deviates from structures of similar size in experimental database. Q-mean Z-score close to 0, the model quality is consistent with experimental structure. Z-score less than 4 suggests potential structure inaccuracies. Local quality assessment, per residue Q-mean score highlight regions with structural inconsistencies such as the loops or the disorder regions. So the models are done. You could also have see that, and as I told, this is the cartoon representation here with the colors make secondary structures. You will get a presentation in PyMol like that. The other one here, these are showing electrostatic interaction, electrostatic surface. You see that when there is red color in the protein, that indicate negative charge. When there is blue color in the protein, that indicate positive charge, presence of amino group. And when there is white color, this shows hydrophobic residues in the surface. And this is the secondary structure predictions. You see the alpha helix, beta sheet and all. It is very detailed and tell you all the interaction presence in the secondary structure. This is other group. And now remember I told you have to do energy minimization. Energy minimization basically is mini simulation, and that's why I keep it here, because today I am not showing you the simulation process which is like very detailed, but in this slide I will talk about how to do the mini simulation. So you have the protein, you have prot.pdb, okay, that is the initial structure, okay. Remember the hydrogens were taken out, so you have to do a common PDB to GMX. Why GMX? Any idea? Because when you are doing minimization, basically it's a simulation. You need to work in a force field. You need to take the force field to calculate out every energy and all terms. So you cannot do a simple program. You have to go to a simulation program. GMX is GROMAX. GROMAX is a package which we are using here. We are not running a simulation, we are just minimizing it. What is the difference between minimization and running a production run simulation? Simulation is a bigger run. Let's say you run for 100 nanosecond, 500 nanosecond, 1000 nanosecond, and minimization is for 100 picosecond. So you need much less computational power and all those things. So when you put it there, you get prot_process.grow. When it comes to GrowMax, all the files would be now .grow. Okay. But along with that, along with the main file, which is .grow, there would be sub files which are postray.itp. Postray.itp is taking the position restraint. What is the position restraint? Position restraint is, suppose you have two carbons, they are covalently bond. They cannot go beyond 1.54 angstrom, right? So they are restrained here. But if they are non-covalent bond, they could move. So a bond could, like two atoms could move from each other or not, that would be defined by positional restraints. That is written here in postray.itp. Another file is topol.top. Topol.top is giving you the idea about topology. What is topology? So basically, when we are talking about atoms, we are just knowing the coordinates, right? Now you are putting a force. So imagine a carbon-carbon bond, two balls of carbon connected with a spring. Now you put a force, this will be going like that, right? So what you are creating would be dependent on the nature of the balls. If it changed from carbon to nitrogen, it will be changed, right? Also it is depend on the nature of the spring. Let's say you put a very hard iron spring, or you put a very small and soft type of spring. They two will behave differently, right? So one would be different behavior of the bond, another different behavior of the spring. Both the things have to be documented here. So you see here, do you see the difference? You are not. So when I show you the first atomic representation, sorry, look at, no hydrogen, right? See, hydrogen is now there, right? You have to see very small things are coming. Then your command is edit conf. So you are actually editing the conformation. And you get prot_newbox.grow. Why you need a new box? Because your protein have to be solvated. You create a real condition. So protein in real condition being solvated, you need to put water. How you will put water? You create a box. See? You can see now? Then you solve it. prot_solve.grow. See here, TIP3P water model. It is not your experiment. So you cannot just, put some water and be. So you have to decide what type of model I will use. There is TIP3P, there is TIP4P, there is other, like totally other, POL5, there is MCDHO, different type of models. What type of models you will give that will either increase your computation time or decrease your accuracy. So you have to be very thinking about. See, change. Now there is water all over. Then you add water. What next? Buffering. How to buffering? add ions, positive charge ions, negative charge ions, right? So you add ions dot MDP and your comment is GROM-PP. The created file is ions dot PPR. Now you add sodium and chloride ion to neutralize the whole system, and it becoming prot underscore solve underscore ions dot grow. See, now you have protein, you have water, but you have this green balls and pink balls, right? These green and pink balls are ions. So what you did, you take the protein, you add hydrogen, then you create a box, then you add water, then you add ions. Then you minimize it. Today I don't go into because it's another thing, but for minimization there are methods. Steepest descent method, conjugate gradient method and Newton-Raphson method. Three methods are generally used for minimization. So you do the minimization and you do the MD run, you get em.grow. Em.grow means energy minimized.grow. You get em.edr, em.log and em.trr as subfile. And you ultimately get energy minimization, energy minimized PDB. See, there is not a lot of difference, right? But few differences. But if you look at here, you see that there are little movements happen, right? Because this is a very standard protein. If you bring a big protein which does not have good homolog, then you do energy minimization and you see a lot of changes. So energy minimization done by GROMAC software package in Linux environment. So the part I am showing you, just by doing that, you could run a energy minimization in the GROMAC software. Then there is Ramachandran plot. So you all mostly know, so I am just passing by saying Ramachandran plot is one of the innovation of Indian scientists where, you know, they have not used a lot of scientific instruments. They have used the bigger version of the what we use in 10+2 geometry boxes. You know, with that and make models out of thermocols. Ramachandran student, you know, I was very fortunate when I was doing summer project in 2000. I got to interact with, I was working with Professor Balram in IISc. So Ramakrishnan, who is actually the originator of making Ramachandran plot, he was mostly retired that time. He used to come and, you know, I was full of questions because I was so in awe about, so I used to talk with him a long time. And I know all these things. He had shown me the models. It's amazing, like in 1960s, taking some thermocols and just measuring with very basic instruments, they come up with a plot which even now with a supercomputer, people cannot, you know, do better. People are doing better means they are making 3D Ramachandran plot and all these things, but the basics, what they had done is really, really good. So in this, what you see the plot regions. Each point on the plot represent a pair of phi psi angle for a single residue. There are allowed region and disallowed region due to steric hindrance clashes between atoms. Not all combinations of phi and psi are possible. The plot highlight, it shows allowed region where favorable conformation, no steric clashes, and disallowed region which rarely observe conformation with steric classes, typically only accessible in special cases. So you could see that you have to go there, PDB sum, and you give the PDB file, you will get this Ramachandran plot. Here you see we have plotted them and I have given the plot statistics for you. I had really done those experiments like this work so that you could see the slides and you could repeat it. At the end, there is conservation analysis with the structure. Remember, they had done wrong structure. So when we are doing up with right structures, you see this is the 3D conservation. So where you see the blue colors, they are variable, nature have made changes there, and where you are getting this maroon color, that means they are conserved. The same thing is here. Another thing I had done, I will very quickly just showing you. I will not teaching you because you guys all tired. So I have given, and you will not get it anywhere other than because I had really worked hard to bring it out. So how the molecular visualization happens started with, the basics to, this is the first structure, first crystal structure representation with metal, and from there to all the representation which are happened in the world of molecular visualization. Then from there I have put the molecular structure, how the molecular structure evolved, how people have done one after one, how the rapid prototyping came, how they have made models, how computer representation, this is the first computer representations of the proteins, and then from there how OTEP came. OTEP is the first software, and how OTEP made by C.K. Johnson get a letter from Kendrew to appreciate that his model is helping him to present his structure. All the history I have noted here. So from there to the modern structures, all of them. So if you are interested, you would, you could go through this Rasmol and then Kaim and conclusion. I have also added the Pymol and Kut because Kut is not there, so I could not have shown you, but I have videos, I included that. Or if you could go to YouTube, search my name, you will get the demonstration of Pymol and Kut. So I will finish here. If you have any question, please ask.
Thank you, Dr. Hajra, and I will like to thank Dr. Hajra with a big applause of his explanatory and a wonderful session. And the session is open for question and answering. If you have any question, you can ask. Dr. Hajra is here to answer you. Question, please.
And you have to do a lot of questioning, please.
Good evening, sir. I have a very small question. Actually, I am very new to this protein engineering. So you told na like.
What is your name? Sorry.
Aditi.
You told.
Yeah. So you told na you are using molecular Caesar for cutting that particular sequencing. So first of all, that sequencing is made up of like purine and pyrimidine sequences, na, like adenine, guanine. So how you will decide like this is the exact point to cut?
No one knows. That's what I told. That's why you have to make these templates, libraries, because this library will help you to go back. It's random.
So you are
This
using
break.
like restriction enzymes
People use restriction enzyme, people use mechanical methods, many methods are there.
Okay, mechanical methods like?
Mechanical just they do tearing.
Okay.
Yeah.
And second thing you told na like,
Restriction enzymes are much sophisticated, but when you are doing it to the whole genome, it's a lot of cost.
yeah, it's very costly. So
Yeah, they are very costly.
purchasing a particular sequence is a little bit difficult. So that's why
That
I'm
is also,
asking.
you don't know, but you don't know that's why you are doing genome sequence. If you know, then why you are doing this genome sequence? So you don't know.
Okay.
See, gene sequencing, gene cloning and genome is completely different ball game. That's why it's very interesting. Every time we do new genome, we get a new plasmid.
Yeah.
Sometime we think like, okay, we'll do 10 genome and then write a paper. Then we suddenly get one genome with, which is creating pandemic. How we know? There is a number called MLST. So you apply MLST and you know that this is a pandemic creator. So you immediately, publish that. Otherwise you will be behind. So we didn't understand one, and we were late for 2 months because we were like analyzing the other genomes and making the paper. 2 groups already reported in between. So genome game is really different, people who are doing science, it's not always even science. It's luck, it's getting a new plasmid, how you get it, how much data you are getting. It's very interesting.
Second thing, the term you have used overexpression in the second second section.
That is our bread and butter, huh?
Yeah. So you are using this buffer solution, or you can say precipitate medium for making that crystals.
Yes.
The purine, like we make usually organic molecule crystals and inorganic
So
crystals.
again, totally different ball game. Like crystallizing a small molecule and crystallizing a protein, like we don't know it will precipitate or it will crystallize. Why it will crystallize. So
Okay. So how you will decide like this particular protein is overexpressing? Like
so yeah, that's a good question. So as I told, we look at a protein. Not everyone do that, but because our lab combine theory and experiment. So what we do, so generally what people do that I say. So you get a gene, you go for cloning. We get a gene, we transfer it to protein. Then we model it, we do a simulation. So when you do simulation, like if I hold this, can you say I hold it strong or not? You cannot, right? But if I do this, now you know I hold it strong. So dynamics makes a lot of things more clear. So we want to put the protein on dynamics, and then we could see that there are some portion of the protein which are really, really weak. No one talked about that, but in the protein world we know that more rigid the protein is, more chance of crystallization
yeah.
and more chance of overexpression. So both are needed for us. So our goal is to make a protein more rigid.
So your first approach is computational.
Yeah, our first
Like
approach is
I'm a
computational.
beginner, I want to do
Yes.
from the scratch. So firstly we will target a particular, our particular target.
Not
first
even
of-
that. So what we do, like, let's talk about my research little bit so you will understand. We work on the area of beta-lactamase. So beta-lactamase are protein which are working with this beta-lactam, penicillin, cephalosporin, all these drugs. So we want to know all the proteins. We want to, like Hajra lab want to work on all the protein. Is it possible? No. Because just for class A beta-lactamase, there are 159 subclasses and 20,000 subclasses. So it is not possible to work experimentally on 20,000 protein. But what we want to work on them. So what we do, we kind of look at their different features and develop first a AI-based model. That AI-based model tells us, it does not tell us that this protein have this function, but it tells us this protein is close to this protein is far from this. So we make a spectra,
Okay.
and we start from this side and this side, and come like that.
Okay,
So
when
that
you-
is our first approach. Then we pick up a protein. We see that this is quite novel and this is publication wise good and all. Then we pick that up, we model that, we simulate that, and we do little chop. Then we get a optimized gene sequence, and that sequence we clone.
So protein you extract from bacteria or
No, we don't
market
extract.
it?
We don't extract.
Okay.
We don't extract. We get the sequence.
Okay.
We are working on the gene sequence.
Okay.
Nowadays, so even when I was in PhD, if you work on snake protein, you actually go to forest and see the snake skin. No, now you don't need anything. DNA synthesis is very, very cheap now. So any DNA in your hand, you could do the sequence, like synthesis of that sequence. So we ask a American company to synthesize that gene. The gene comes, then we work on that.
Okay.
So the gene which comes here, this is not the original one. But then sometime the reviewer challenge. Then we also give the original one. Otherwise, we try to make the protein rigid.
Okay. And my last question, being a spectroscopist, is there any scope of like fluorescent molecule collabing with your work?
Obviously, so there are
Like
so
labeling kind of thing is possible with
yeah.
fluorescent dyes?
So I didn't talk about. I have a company which work on device. So my dream is to develop number, especially kind of for girls. When you travel, you get bad restrooms and then get inflammation. You probably get medicine, right? I don't want any of them, any of you to get a medicine. So my dream project, which we are kind of close to, we have already done 8 patents. So if you go to hospital and any sample is given, now within 1.5 hours, we could have tell you, don't take these drugs, take this, don't take these antibiotics, take this antibiotic. My dream is number. Nowadays you hear about pollution index, right? It was not there probably in your childhood also, right?
Yeah.
My childhood, I never heard about. So it is possible to develop some numbers. So you have a small device, put like small like spit or urine, and you get the number. You are happy that your AMR index is not serious enough. So that will change the way we see the medication happen. So many people are taking so many bad medicines. Not like stopping people from taking medicine is probably the best way to work in antimicrobial resistance. So that, as coming back to your answer, so we started with a colorimetric compound. Then we shifted to a fluorescence compound. Then now we are working on a luminescence compound. We are doing a straight. So we are doing a straight couple.
Okay.
So we work with their lab, Sandeepan's lab. We work with a DRDO lab where they are a lot of body pies for us, body pie extract. So we do a lot of this. See, like personally, I think you should open your research. It's not that I know these techniques. It's where the problem is. So any of the techniques which fits to solve my problem is always, good to collaborate with.
Okay. Thank you, sir.
Good afternoon, sir.
Good afternoon.
Sir, I am
What is
Manish.
your name? Sorry.
I am Manish Sharma, sir. I am
Manish.
Manish Sharma. I am doing MSc here, sir. Sir, by the way, sir, it's a hypothetical question, but I want to know that by the replication of DNA of some flower plants,
and bacteria like Klebsiella pneumia, which can emit fluorescein at all the 7 extinction wavelength. Can we go with the existence of these types of plants which can also emit fluorescent?
Repeat.
Okay, sir. Sir, by the replication of DNA of some flower plants and bacteria, like which can emit...
When you say flower plant and bacteria, what do you mean there?
Sir, DNA of flower plants and bacteria.
You want to breed?
Yes, sir. And by the replication of these DNA, which can, sir, bacteria like which can emit fluorescein with all the extinction of 7 wavelength. Can we make plants like this?
But इसके लिए breed करने का क्या जरूरत है? When you know protein, you just like see. I don't know much about biology. I'm learning, but I don't know that breeding is possible or not. But as you say, the gene is definitely possible to pick up and insert on the like transform on the bacterial cell if you want to express.
Okay, sir.
That is always possible.
Okay, sir.
But breeding part I don't know.
Okay, sir. Thank you.
Any other question?
Good afternoon, sir. I am Yanus Mullah. I do have a MSc.
और एक बार बोलो नाम.
Yanus Mullah.
अच्छा.
I do have a MSc. My question is, what is the property and relationship both viral genetic materials and bacterial genetic materials?
What is the difference you mean?
Properties and the relationships.
Relationship is a like kind of not suitable word here, but there are many differences. The major thing is when you think about a virus, they are much smaller organism. So, and they mostly use your system when they are coming inside you. So, when you target the genes, the number of genes are much more in bacteria. So, there are much better possibility of getting a gene which is totally different from you. But in case of virus, overall you get 10 gene or 12 genes which are coming from virus. That's the main, problem. All those, if you see this flu and all, we don't have any medicine. That's because of that. People used to let them go, come and go, but they never create such pandemic what they have done in COVID time. Before that they used to come, they used to like put fevers and go.
Thank you, sir. One small question. What is the difference between plant and animal proteins?
As such, there are different proteins, right? So you have to talk more specific about, there are many common proteins. There are many common proteins. All the conserved proteins are common. Then for lifestyle, there are differences. So it's better to think about why this protein or how this protein is different rather than how many proteins are different.
Thank you, sir. Once more, sir.
No problem.
How do the work DNA RNA replication inhibitors and when it's working time?
I was talking about this nucleoside analogs. Those are actually a part because they basically like stop DNA synthesis and all. Those are part, but nucleoside analogs, and nucleoside analogs are of many types. So they are one. Then there are other, class of drugs. Again, like the thing you are asking is a very, like it's targeting a very broad area. You have to be little specific where you are asking the question. Because there are so many different type of, inhibitors which are used as like in this inhibition process. As today I have talked about one. There are many such classes.
Thank you, sir.
Yes, here is a question.
Good evening, sir. This is Nikita. Sir, I was having a question related to cancer. When we study how cancer takes place, it is just due to the difference in codonic sequences. The codons get addition ho jata hai and deletion and all. So, is it possible that we work on how the proteins get disfigured due to these codonic replicancies and all, and we just focus and we study all these proteins and we create models to work on, and using those models we can find some molecular sieves and all.
You speak, you speak.
So that we can just exclude that part of the protein that is causing that cancer, due to which the cell is behaving abnormally.
Any change in protein will not help us there. If you want to make a change, you have to make change in the DNA level, first of all. Second of all, you started with that these happen in cancer. No, not. There are many of the reasons. This is one of the reasons where there is a point mutation. The problem of cancer is if the cancer comes to blood. Why? When it comes to blood, it spread to the whole body. Otherwise, cancer is so curable nowadays. So the problem is not the mutation. The problem is spreading the mutation. When it goes to
mutations are taking place at a place, so if we can control that mutagen.
No. Yeah. But that's what I'm saying. Be it a mutation, if we could see when people are detected at the initial level, most of them get cured. The problem is when it starts spreading. So what happened? Let's say what you say happens to gene A. Now gene A creates a problem that makes the cell division faster. Because of the faster cell division, uncontrolled cell division, there is tearing. No, there is tearing. When there is tearing, a lot of genes are getting mutated. So when doctors say there is nothing we could do, it's not one mutation left, or it's not one protein affected. It's let's say 10-20% of the protein of the body is getting mutated because of this process.
So is there any way that this molecular docking and protein structuring can help us out in this?
Yeah, definitely. But the thing is, whenever it identified in primary level, a lot of things are done. As you see, I started with the nucleoside analogs. It's a great example. Nucleoside analogs are involved in so many regular chemotherapies. A lot of people's lives are saving because of that. The problem is not that. The problem is two problems. One, breast cancer. One example. I had a family history of a lot of my very near ones. So they identified at the age of 30, 25 to 30. That time chemo is given, but it's a bulk. So somehow one cell left. So it's a common history in our family. Cancer detected, chemotherapy and all done, patient is good. It's come back after 30 years, 39 years, 42 years. So that another one problem.
So that is the problem, that these chemotherapies and all the therapies that we do right now are not 100% effective, and there are cells that are left that have these.
Yeah. So it's not that they are not 100% effective. It's that what type of cancer is. It happens mainly in breast cancer because of the lump of the tumor. The tumor generally become big. So even if it is amputated, there is some
Some of the
seeds
cells are there.
staying there, and they don't, like, somehow they get resistant.
Is there any more questions? If not, please join me in thanking Professor Hajra for this wonderful lecture. And I believe you had a very good hands-on sessions in this particular time, and you learned a lot from him and asked so many questions. So it's my pleasure from the organizing side to have a token of appreciations. Okay, thank you very much. So we'll close today's session. But some announcement, tomorrow we'll start at 10 o'clock. So please be here 10 o'clock, because tomorrow we have to spend some time on the valedictory sessions. Tomorrow if you're not coming on 10, I'm sorry, I cannot give the certificates. That's final. Because that will be given on the stage, you have to keep some time, you have to give the schedule accordingly. Okay, so please be on time, please. Tomorrow 10 o'clock we'll start. Okay, thank you very much.